ELISA, or enzyme-linked immunosorbent assay, is a type of immunoassay that is commonly used to quantify levels of a specific target within a sample. Samples routinely used in ELISAs include serum, cell lysate, saliva, cell culture supernatant, tissue lysate, and urine. ELISAs are usually run in 96-well or 384-well polystyrene plates, which passively bind antibodies and proteins. Having the reactants of the ELISA immobilized to the microplate surface makes it easy to separate bound from non-bound material during the assay. ELISA can have different formats. The four main types of ELISA are direct, indirect, sandwich, and competitive. Each type of ELISA has its own advantages and disadvantages. Direct ELISA. In a direct ELISA, an antigen or sample is immobilized directly on the plate and an enzyme-conjugated detection antibody binds to the target antigen. Substrate is then added, producing a signal that is proportional to the amount of analyte in the sample. Since only one antibody is used in a direct ELISA, they are less specific than a sandwich ELISA. Direct ELISA has the advantage of being a fast and simple protocol. However, it is less specific since we are only using one antibody. Additionally, there is a potential for high background if all proteins from a sample are immobilized in the well. It is expensive due to the use of a labeled primary antibody. Labeling may affect the affinity of primary antibody to the antigen. Indirect ELISA. An indirect ELISA is similar to a direct ELISA in that an antigen is immobilized on a plate, but it includes an additional amplification detection step. First, an unconjugated primary detection antibody is added and binds to the specific antigen. A conjugated secondary antibody directed against the host species of the primary antibody is then added. Substrate then produces a signal proportional to the amount of antigen bound in the well. In an indirect ELISA, signal amplification is achieved by using the secondary antibody which binds to several epitopes of the primary antibody. An indirect ELISA is versatile and the same secondary antibody can be used in many experiments with different primary antibodies generated in the same host. Different detection methods can be used with the same primary antibody, such as colorimetric, fluorescence, chemiluminescent, etc. The disadvantage of an indirect ELISA is that there is a potential for cross-reactivity caused by secondary antibody. Sandwich ELISA. Sandwich ELISAs are the most common type of ELISA. First, a capture antibody is coated on a microplate, followed by sample addition. The antigen of interest then binds and is immobilized on the plate. A conjugated detection antibody is then added and binds to an additional epitope on the target antigen. Substrate is added and produces a signal that is proportional to the amount of analyte present in the sample. Sandwich ELISAs are highly specific, since two antibodies are required to bind to the antigen of interest. The advantages of sandwich ELISA are that it shows the highest specificity and sensitivity among other ELISA platforms. It is also compatible with complex sample matrices. The disadvantage of sandwich ELISA is that it involves a longer protocol which may be challenging to develop. Comparative ELISA. Comparative ELISAs are commonly used for small molecules when the antigen of interest is too small and therefore has only a single epitope or antibody binding site. In this technique, the antibody is first incubated with a sample containing the antigen. Now, a microtiter well is coated with antigen and the antigen-antibody mixture is added. The more antigen present in the initial solution phase sample, the less free antibody will be available to bind to the antigen-coated well. After washing off the unbound antibody, an enzyme-conjugated secondary antibody specific for the isotype of the primary antibody can be added to determine the amount of primary antibody bound to the well. In the comparative ELISA, the higher the concentration of the antigen in the original sample, the lower the final signal. This technique is usually used to quantitate small molecules such as hormones, peptides, or drugs.